All right, hello everyone. Welcome to the CMap Links and L1000 workshop. It's a little bit after 10:30, so I think we'll get started. I noticed there are a couple of questions in the chat about whether this would be recorded, so we are going to record it, and we will post a link afterwards so everyone can have access to the video. So why don't we get started? First of all, thanks everyone for joining. My name is Ted Natoli. I'm a member of the CMap team here at the Broad Institute, and myself and a few colleagues will be taking you through a workshop today and tomorrow. The focus for today is going to be around data access. So we'll start by giving some background on CMap and the L1000 assay and a previous data release. We'll talk about our updated data release, which now contains over 3 million gene expression profiles. And then we'll focus on some data access modes, including a migration of our data store to Google BigQuery. And we'll show some interactive demonstrations around how to access the data through some web applications, static downloads, and then a little bit more of an interactive session using a new toolkit we've developed in Python to enable access to our data in BigQuery. And we'll try to reserve about 15 minutes at the end of the session for general Q&A. So hopefully you got an email from us earlier today with some instructions on things that you'll need for today, but just to recap that, it'll be very useful if you have an account at clue.io, which is our main website. You can go there to sign up if you haven't done that yet. You also will probably want a Google account. We're going to use Google Collaboratory to share notebooks with you. And also that's where our data is stored. So that'll be a useful thing to have, although it's not absolutely required for today. And mentioning those notebooks, we have a GitHub repository, which should be publicly accessible at this link here, where we have some notebooks that we'll be walking through. You don't have to download them. You can if you want to, but we'll be using Google Collaboratory to walk through them interactively, so you can just launch them right from your web browser if you like. So you can sort of participate as much or as little as you want. I think you'll probably get the most out of it if you kind of try to follow along with us, but of course it's fine to just sort of just watch as well and participate that way. In terms of getting in touch and general participation today, so we have our website at clue.io, which is a great place to start. And that has an FAQ section called our Connectopedia, which has a bunch of articles that we've written to kind of help folks out who are new to using the resource. We also have weekly office hours that we host via Zoom, and you can visit that link there to join. And you can also always email us at clue@broadinstitute.org, and we'll try to get back to you as quickly as we can with any questions. But for today, please try to use the Zoom Q&A function to post questions during the workshop. You can use that as often as you like. We'll have someone monitoring that during the workshop. You can also use the chat function if you want to communicate with each other, but please, if you could try to pose questions to the panelists using the Q&A function, that would be great. And as I mentioned earlier, we're going to have a Q&A session at the end to try and answer any other questions. So to give some background about the NIH Links Consortium, this is a program that the NIH started several years ago now. It's called the Library of Integrated Network-Based Cellular Signatures, or LINKS for short. And so we at the Broad are the Center for Transcriptomics, but we're one of many labs across the country who are working to build a resource of perturbational, high-dimensional cellular readouts that can then be interrogated to try to advance biomedical discovery and hypothesis generation. And we work closely with the LINKS DCIC, the Data Coordination and Integration Center, to make our data publicly accessible and analyzable to the community. So the main idea behind the connectivity map is that we want to use mRNA-based signatures of cell state to link disease, drugs, and genes. And so what that means is that we use the mRNA expression state of a cell to sort of capture information about what is happening in the cell, and then we can compare those fingerprints of gene expression to each other to find associations between drugs and genes or disease based on similarities or differences in their transcriptional state. So the experimental system that we use is we culture human cells in 3D 4-well plates, and then we perturb them with some type of reagent. Could be a small molecule or an shRNA or CRISPR or really anything. And then sometime afterwards, we measure the downstream transcriptional changes that are induced by that perturbation. And we capture those in a database of these mRNA expression signatures. We can then compare those signatures to each other or to external data to try and derive hypotheses about relationships between the perturbations or between the perturbations and other cell states of interest. And so to make this as useful as possible, we realized that we needed to scale this up to millions of expression profiles. And so other contemporary assays such as RNA-seq or a few years ago, things like Affymetrix, they just didn't scale quite as well. And so to facilitate this, we developed a new assay called L1000, which is based on the Luminex system, where we're measuring only a subset of the transcriptome of about 1,000 predetermined genes that are representative of the remainder of the transcriptome. And then using those 1,000 landmark genes, we then computationally infer about 11,000 remaining transcripts to get a more complete picture of what's happening to the transcriptome. And so this assay, when operating at scale, can generate around 200 to 384 well plates a week. And so this, coupled with the lower cost of using a system like Luminex and not measuring every last gene, enabled us to generate the CMap resource as it exists today. And so with that assay, and with some algorithms that enable comparisons of these gene expression signatures to each other and to external data, that sort of comprises the previous version of the CMap links resource. It was about 1.3 million gene expression profiles across about 28,000 different perturbagens and around 80 different cell lines. And this was released back in 2017. And so there were sort of two large partitions of this data set, as the graphic in the center illustrates. The one on the left is what we call Touchstone. That's sort of the main reference data set where all the perturbations are well annotated in some way. So if it's a small molecule, we know something about the target or the mechanism, or if it's a genetic perturbation, we know the target gene and it's the wild type version of the gene, and we know what pathway or what gene family it belongs to. And so comparing external gene sets to those perturbations enables a hypothesis development, because you have some idea of what your query gene set sort of looks like in terms of what's known. So of course, that was a few years ago, and we've been making data all the while since then. At this point, we've got around 3 million gene expression profiles that in total. So this new resource contains some data from a variety of different data generation efforts, including the Links Phase 2 efforts, as well as other academic and commercial partnerships that we've engaged in. And notably, the data set is now expanded into different types of perturbations and cell lines, including hematopoietic cell lines, some additional non-cancer models, and notably CRISPRs, which we did not have any of in our previous data release. And as I was saying, that release now is over 3 million gene expression profiles and a little over 1 million signatures when you look at replicate collapsed signatures. And so this plot on the right just gives sort of a very, very high level overview of some of the characteristics of the data. These are distributions of the signature strength in terms of how many, what proportion of the landmark genes are significantly modulated by a certain type of perturbation. And so you can see even there, you can see some differences between the perturbation types. For example, compounds seem to be the strongest modulators. They sort of have the longest tail out to the right, where CRISPRs seem to be a little weaker overall. There's nothing terribly insightful from that, but it just gives you some sense of different patterns that you might see from a large data set like this. To give a little bit more detail about what's in the new data set, these are some stats about the different types of perturbations that we've profiled, the different cell lines and genes and so forth. And these graphs compare the 2020 data release to the 2017 version. So there are some notable changes here. For example, we've now got over 11,000 CRISPR reagents in this new release, whereas we had none back in 2017. And there are about 14,000 additional compounds that we've profiled relative to the last data release. This plot here shows the number of cell lines by lineage that we've profiled in the new data release relative to the old. So there have been notable increases in areas of lung cancer and hematopoietic lines like lymphoma models and leukemia as well. In terms of compound mechanism, there's about 100 or so additional mechanisms that we've now have representation for in our data relative to the last release. And in terms of genetic perturbations, there's about 1,200 genes that we've profiled by each of shRNA, CRISPR, and cDNA overexpression. So this just gives you sort of a sense of the data that's available and may already start leading into some questions that, that might be interesting to ask of this data set. If we focus in on compounds, we've also sort of expanded in the dose and cell line dimension for a number of compounds. So these two figures indicate the number of compounds that have been profiled in at least a certain number of cell lines and at least at a certain number of doses. And so you can see there's over 1,000 compounds, well over 1,000 compounds that have been profiled in 20 or more cell lines and or at least 12 doses. So there are some where the data is sort of has very deep coverage across those two experimental dimensions, which we didn't really have in the previous release. And so that sort of leads into a few interesting subsets of data that I wanted to highlight just very briefly. So as I just mentioned, we now have some compounds that have been profiled at high numbers of doses. So in particular, there's a subset of 1,500 compounds that we profiled at a fixed set of 6 doses. There are now CRISPR knockout signatures for over 5,000 genes and 10 cell lines. And we have 140 compounds that have been profiled in 11 hematopoietic cell lines. And a few examples of these data sets and questions we might ask of them follow. So for example, for compounds, we might now ask, do we see a dose response? And this is sort of a very simple way of looking at dose response, but the idea here is to ask whether the overall transcriptional activity, which is assessed as the number of genes that change, and also how correlated are the biological replicates for a particular compound dose combination. And we can compare that activity level to the dose. And so we can see some interesting patterns that emerge for a few example compounds. For example, there's this compound here, AZD5438, which I think is a CDK inhibitor, that winds up being broadly dose responsive. So in all the cell lines in which it was tested, it gives a pretty consistent dose response. The signature gets stronger with dose, opposed to a compound like bortezomib, a proteasome inhibitor, which is sort of universally strong at all doses. In most cell lines, in fact, in all the cell lines. Whereas norepinephrine, which I think is an estrogen receptor antagonist, that is selectively dose responsive in only one cell line. And fulvestrant, another estrogen receptor antagonist, is selectively dose saturated in only one cell line. So even just looking at something as simple as the overall activity level of a signature and comparing that with dose, you can see some interesting patterns that emerge. If we take a look at the CRISPR data set, this heat map gives us sort of a high-level view of the transcriptional activity of about 5,000 genes in 10 different cell lines. And so you can sort of take slices out of this heat map to look for genes that have interesting patterns. So here's a couple examples of context-specific patterns, genes like estrogen receptor and BRAF, where the signature is notably stronger in a subset of cell lines where that receptor is expressed. As compared to genes like MYC or a proteasome subunit, which seem to be generally strong across most cell lines because they are central to all of these cell lines. So there are, even at this very high level, some interesting patterns that emerge. And taking a look at the hematopoietic data set, we focused in on a compound class called BTK inhibitors, a Bruton's tyrosine kinase, which is only expressed in hematopoietic cell lines. And then drilled into the hematopoietic cell lines that we profiled, in which there are a few different subtypes of diffuse large B-cell lymphoma, including the ABC and GCB subtypes. And the plot on the left is just a principal component analysis of the DMSO samples from this data set. And even there, we can see that at baseline, without any compound treatment, we can see differences between the different DLBCL subtypes. And if we look at the BTK inhibitor class, which I think there are 11 compounds in that class, this plot on the right shows the overall class coherence of those BTK inhibitors in each cell line. And so the further out from the origin you are, the more coherent the members of that class were in the given cell line. And so interestingly, we see that in the ABC subtype, the class is much more coherent than in the GCB subtype, which tracks with the clinical observation that the ABC subtype is more responsive than GCB to BTK inhibition. So interestingly, we can see these patterns that are reflected in the L1000 data for this particular class. So in addition to adding additional data to our resource, we've also been taking advantage of the crowd using these public crowdsourced competitions to improve our data analysis pipeline and other algorithms. And so over the past 4 years or so, we've ran 4 of these contests to improve different aspects of our computational pipeline. They've all been quite successful. We've actually got one that just concluded the end of November or early December, where the challenge was to use CMap and cell viability data to predict compound mechanism of action. And so we're still just digging into the results of this, but it was quite successful. We had over 5,000 competitors, and the top solution was around 20% improved over a sort of a naive benchmark. And so, these have been quite interesting for us, and they serve as a way not only to improve our algorithms, but also to engage folks who are outside of the biomedical research community and bring them into the fold and get them involved in our work, which has been quite exciting. So there's a lot of discussion about the data there, but I think probably it's very useful to talk about how to actually access this data. So we've recently migrated our data over to Google BigQuery, which is a highly performant data store hosted by Google. And this is sort of not a total transition from, but an augmentation to the previous methods of storing data, which were in flat files, text files, and GCTX files, which some of you might be familiar with, which are essentially annotated data matrices. And so we've worked out a process to take these flat files in GCTX matrices and convert them into a relational database schema, which can then take advantage of indexing, controlled vocabulary, referential integrity, and so forth. And then load that over into BigQuery, which makes this data available to things like our Clue APIs, and of course, external users such as you all. So this enables really efficient data access to arbitrary subsets of the data. So you don't have to download the entire data set anymore, although there are some cases where you might want to, but there are many, I think we can serve many use cases without needing to do that. And it also allows us to do more continuous updates. So we can get new data out into the public domain faster using this method. And so we've developed some tools on top of this BigQuery data store to help facilitate data access. So we'll go over each of these, but there's a metadata browser app on clue.io, which we'll show in a minute. We also have a code library in Python called our CMap BigQuery or CMap BQ toolkit, which we'll also demonstrate today. We won't be showing this today, but it's also worth mentioning that BigQuery has its own console, and Google has their own developers toolkits that can also be used to access this data, although that's not something that we've written or directly support, but that is another option. And of course, you can also still download the data in GCTX format on Clue, and eventually from the DCI website and from Geo as well, and we'll show how to do that today. And these tools all serve sort of different use cases. And so these are just a few examples of types of questions that people might ask and the corresponding tools that we think would be best to answer those questions. So for example, if you wanted to know which compound mechanisms have been profiled in the links data set, you could just get a list of those by using the metadata browser, which I'll show in a minute, or by using the CMap BQ toolkit. So there's no need to download anything. You could use either of those utilities to just figure out what data exists. If you then wanted to get, let's say, all of the level 5, so this is our replicate collapse differential expression data for genetic reagents that target the gene KRAS, that's not a large number of signatures, so you could use the metadata browser for that or the BQ toolkit, but you wouldn't need to go and download a giant GCTX file just to get the data for KRAS. On the other hand, if you wanted to take all of the data, take all 1 million signatures and do something like train a deep learning model to predict compound target, then you might actually need to download the data. Things like the browser app and the toolkit are not well optimized for streaming large chunks of data. And so in that case, you may want to actually download it. So it's sort of worth thinking about your application and your use case before you go ahead and just download a giant file. And if there's any questions around that, we can certainly help try to figure that out with you. So that, I think, will take us into the first demonstration. So the first thing that I'll show is how to use our metadata browser app and our static downloads on clue.io to access the data. So I'm going to get out of my slides and just head over to clue.io. And so just to give a sort of a brief overview for folks who haven't used Clue before, this is the homepage, and there's a few things you can do here. So there's a search box in which you can type the name of a gene or a compound, and it'll take you to information that we have about that gene or that compound. So for example, if I just search for MYC, this will give me a list of the genetic and compound reagents that we have that target the gene MYC. You can also search for compounds or cell lines or other things like that in this from the home page. But what I'm going to show is a specific tool that's under the tools menu. So most of the tools that folks will be using are under this tools menu. We'll go over a few of these additional tools later today, but the first one that I'm going to show is the data library tool. So the data library is a location in Clue where we store different data sets that users can access. So my version of this, since I'm logged in now, may look a little different from yours. I have a few more projects that I can see, but the one of note for everyone is this publicly accessible entry called the Expanded CMap Links Resource 2020. So if I click on that, there is one data set that lives under this entry, and I'll click that. And that takes me to this page where there are links to download a number of different files associated with our new data release. So I think it's worth just mentioning at this point that this is in some ways a beta release of the data, in that we are still sort of in the process of vetting the data and working out the experimental, not experimental, but the modes for enabling access. There may be some tweaks that we make as folks use the data and we discover things that need to be fixed. So it is beta in some ways, but we do have a versioning system in place, and so any updates will be reflected here in the file names, and that'll all be handled in a clean and understandable way. But just to give a sense for what all is here, we provide the CMap data at each of 3 data levels, so levels 3, 4, and 5. And if there's any questions about what those data levels mean, there's some information here about what that all means. I won't cover this today, but if there are additional questions, we can try to address that in the Q&A. But there's one file for each data level and each perturbation type combination. So for example, level 5 CRISPR signatures are in this particular GCTX file. And so it's split up this way to enable faster downloads, because making it all one large file would be pretty onerous to download. And even still at this size, some of the files take a bit of time to download. So I just encourage folks to think about exactly what their use case is and whether they really do need to download the data or all of the data. And as I said earlier, we can certainly help you guys try to figure that out. There's also associated metadata. So each of these data matrices is a GCTX file. It is not annotated, but the annotations are provided here in separate files. So for example, there is a siginfo file, which has all the metadata for the level 5 signatures. So the entries in this file correspond to the columns of the level 5 data matrices, as well as a cell info and gene info file, where gene info gives you the metadata for the features of the rows of these gene expression matrices, and cell info tells you information about the cell lines in which the experiments were done. There's also an Excel file here that contains information about what all the field names in each of these different files mean. So you can kind of decode that and see in plain English what each of these fields means. So this is the place to go to download data. But if you want to do something a little more interactive and kind of explore the data a little bit more, we have our new metadata browser app, which provides you an interface to write queries in SQL to interrogate the metadata. So there are two modes of querying the metadata at the moment. Right now you can query drugs and signatures. I'll start with drugs just to give an example. So we can just write in a SQL query, and there's one that's pre-populated here, where this query is asking for the ID, name, mechanism, and target and structure from our compound info table, where there is a target and a mechanism annotation. So if we submit that query, we get back this list where there's one entry per compound and annotation combination. So you can see there are some compounds that appear more than once because they have more than one target. But this gives you the information we have about these compounds. And of course, we put a limit here in the test query of 10. However, I can just get rid of that and run the query again. And this will take a minute to run because there are many, many of these results. And you can see this updated here, and it said it found 1,000 results. And so you'll note that we limit the number of rows returned to 1,000. And that is to just limit the amount of data that people are pulling out at one time, because it can wind up being, you can have some performance effects when returning much more data than that. But for example, if you wanted to know how many of those compounds exist, you can alter the query to ask that. So I only saw 1,000 in this table, but if I wanted to know how many there actually were, I can just change the query, and this will take a second to run. But there are 3,100, a little over 3,000 compounds where we have a target and an MOA designation for that compound. And, you know, for examples like this, you can export the table if you want to. There's a few other types of queries that may be interesting. And so this kind of gives you a sense of what data exists for compounds. But if you wanted to actually get at the data itself, you can then query the signatures for these reagents. So this example query is asking for a few different fields from our level 5 signature annotations table where the perturbation name looks like chloroquine. So let me run that. This will take a minute to run. So we've got 413 of these signatures here in this table. This is their metadata. If I wanted to, I could download this by clicking the export button. And I'll just save that to my downloads folder. And this just gives you a text file with all of these annotations that you could use. Even this has some analyzable data in it. For example, we do have the replicate correlation value and the transcriptional activity score, so you could run some analysis on that in and of itself. But if you wanted to get the signatures themselves, the level 5 data corresponding to these signatures, you could do that by clicking the query data matrix with sig IDs button. So I'll do that in a second, although I just wanted to show a couple other examples here. So if I wanted to find, for example, all of the compounds that, or all of the signatures in the cell line A375, I can just paste that in, and this will take a second to run. And now again, my results are getting clipped to the first 1,000. So if I wanted to know how many there actually are, I can then alter the query to count the distinct signature IDs in the cell line A375. And this will take a second to run again. And now that returns a count of over 90,000 signatures in the cell line A375. So that just gives you a sense for what all data is there. We want to go back to the chloroquine example to illustrate how to actually then get the signatures. I'm going to alter the query a little bit. So I don't want to pull all 400 signatures. I'm going to then restrict these signatures to the exemplar signatures for chloroquine. And so without going into a ton of detail, that just means that for each perturbation and cell line combination, we've designated a signature as the exemplar. And that is the one where the data quality was the highest, and we saw a good consistency between replicates. And that's encoded in this is NCS exemplar field, which is just a Boolean with ones or zeros indicating where that signature is the exemplar. So if I run that again, I should now get fewer results. So there's 45. 45 signatures where there's a single signature for each compound cell line combination. And so now I am going to fetch the data for these signatures. I want to name my query something so that I know how to look it up later. And I'll hit query the data matrix with sig IDs. And so in a few minutes, these results appear in the history app. I'm going to just jump over to that now because I've ran this earlier today. And so the result here is already pre-populated. So in my history app, this actually, just to give a brief walkthrough of this keeps a record of all the analyses that you run through Clue. So I've got a bunch of analyses in here that I've done over the past several months. This is the most recent, which is a request to fetch the signatures for chloroquine and chloroquine-like compounds. So I can highlight, this is the one I just submitted, but this is an older one. So if I click the button, the check marks next to this signature, I can then come over here and click View in the heat map. And that's going to bounce me over to this application called Morpheus, where I now have all of the signatures. I'll just get rid of this. For these compounds in all of the 45 signatures that I requested. And so what this view gives you is a matrix where the columns are the different signature IDs and the rows are all the different genes that L1000 reports on. So there's a little over 12,000 rows and 45 columns corresponding to the signatures that I selected. So this app is sort of an interactive heat map viewer. And I won't go into all the details of what all it can do, but you can do sort of simple things like sort the rows and columns by clicking on an annotation field. So if I want to sort it by compound, I can do that. I see there's chloroquine and hydroxychloroquine are the two different entries here. You can see the compound TAS, the activity score for each of the signatures, which I can then sort by clicking on that field. And now this is the strongest of the signatures. And if I wanted to see what are the genes that are most modulated, I could sort this by double clicking on that column, and it then sorts all the rows in descending order. So these are the genes that are most affected by chloroquine in this particular cell line. And you can do a few other things too, like if you wanted to save this data set as a GCT file, you can do that, or save an image of the heat map, you can do that as well through the file menu. You can also do things like sort and filter the data. So for example, if I wanted to now just restrict this data to only landmark genes, those that are directly measured, I can add a filter and just restrict it to landmark genes. And now you'll see that there are only 978 out of the 12,000 rows that are being shown. These are only the landmark genes. And I can then, if I want to make that into its own heat map with just this particular signature, I can do that. So now I have just this strong signature with only the landmark genes, and then I could then download this or download an image of it if I wanted to. So there's a few more things you can do here with this Morpheus application. I won't go into all of it, but it's worth exploring for folks who haven't used that before. The other thing I'll do just quickly to illustrate is if I go back to my history app, if I don't want to view this result necessarily, I can just download it by clicking this little download button, and this will save a zip archive of that data to my desktop. And let me just share my full screen so you can see that. So within this zip folder, once I extract it, I've got my GCT file, which is the data matrix that has all the L1000 signatures in it, as well as gene and sig info for row and column annotations. So you can then go take this, these files and do some other analysis with it, or kind of use them however you like. So I hope that gives a sense for how to explore some of the data and access smallish subsets of the data via Clue. I'm then going to transition over to my colleague Anoop, who is going to talk a bit about how to access data using the Python CMap BigQuery toolkit that he's written. So Anoop, I think I'm going to stop and hand over to you.
That sounds good. Can everyone hear me? Ed, Rajiv, can...
Yeah,
Yep, we can hear you, Anoop.
Okay, great. Yeah, as Ted said, and kind of to answer some of the questions that have been appearing in the question and answer section, I'll be going over how to access some of the data that's hosted in BigQuery from a Python kernel or a Python interface, such as Jupyter Notebook, using a toolkit that I've created called the CMap BQ, or which we call the BQ toolkit. So this is meant as a way to retrieve targeted subsets of the data, as a few people are asking questions about. So we'll format this in a few ways. So I'll introduce you guys to where you can find some of the documentation, and then there are a few notebooks we have, a tutorial notebook and then a demo. So we will first go over the tutorial, where we're hoping it'll be a little bit more interactive, and you guys will be able to try to run these on through Google Colab, as well as there's also a more demonstrative notebook where we'll walk through some potential uses of the data and go through some example workflows for analyzing data. So just to start off, we will want to navigate to this link. This is where the documentation is for the CMap BQ package. So this is just CMap BQ.readthedocs.io, which is where we host all of the documentation. And then from here, you want to go to this link using CMap's BQ toolkit. So within some of the sign up emails sent out, there was kind of a request for people to go through this credential setup. The toolkit does require a Google Cloud account. And then from that Google Cloud account, a service account and JSON key are necessary to run this. For this workshop, we've created a way to, we've created a credentials file that'll be available to download in case you haven't done it, but I'll also walk through how to upload it into Colab and get it up and running. Because the workshop credentials file, we will actually deactivate after this workshop. So you will have to follow these credential setup instructions to get this key. But so just to start, what we want to do is go to that page and then click right here are links to the tutorial notebook and demo notebook. So we'll start off with going to the tutorial notebook. So just a quick introduction, this is Google's Colab. This is essentially like a Jupyter notebook hosted on Google Cloud. So this is linked also, and this is also available through the GitHub repository, which we will send out links to in the future. So again, the CMap BQ allows for targeted retrieval of relevant gene expression data from the resources that we've put up on BigQuery. So just to expand a few things, this tool is available and installable via pip, so it's hosted on PyPy, and can be installed just by running this pip install CMap BQ. These are flags just to make sure that we have the most up-to-date version. So I guess first I'll start off and just wait a few moments for people to catch up. I think if you can navigate to this page and then try to run this cell. So these are all cells, just like a Jupyter notebook. And you can run them just by clicking this little play button to the left of the code. So here we'll run it. It talks about it not being authored by Google, but it is available in open source on GitHub. So we're going to just say run anyway. And here it's
Anoop, I'm sorry to interrupt. Is there?
Yep.
Do you think you could zoom in a little bit? The font is a little bit small for folks to see.
How's this?
Thanks.
Is this better? Okay.
Thank you.
Thank you. So yep, first we'll just run this pip q install. Excuse me, I just want to reset this so that I'm starting with you guys. So we'll run this and this will install the package from PyPy. And then we have a few standard imports such as pandas and Seaborn that allow us to do simple things like plotting and creating data frames. And then the next step we want to do is if you did go through this credentials setup, this is the moment where you're going to want to uncomment this credentials file path line and then go to the files page and then select upload, upload file. So here, this is an upload file. And for me, for example, I have this BQ credentials JSON key. This is a key that just authenticates you with Google Cloud. So now I've been able to upload my credentials file into Colab, and I can copy this path. And then we just want to paste the path right there, right? So what this block of code is doing is it's importing the CMap BQ packages. Some of the main packages are the query package, and then the config package is the package that helps us set up the credentials. So if you're running this locally, this setup credentials file or function only really needs to be run one time. It will set up a configuration on your machine. But otherwise, in this Colab environment, we're going to be running this. And then there's a function to get the BQ client. So the client is an object that we're going to need to pass to every function that we use. And it is essentially what we'll be requesting from Google BigQuery whatever request that we're making for the data. So here we'll just run this. And then we should have a client object. So alternatively, if you don't have a credentials file yet, for this workshop, we've hosted one on S3 that should be accessible for everybody. So you can just uncomment this line, this block of code, just so that so you're able to interact and follow along now. Again, I'll say this file will be deactivated, so it won't work, in the future after this workshop. But so people can follow along now, we can run this block of code, and then make sure this is commented out so that the credentials file path is the correct one. And then we can, again, run this, and it should authenticate us and give us a BigQuery client. So I'll kind of pause and let people catch up with that. And while people are running those few blocks of code, feel free to any issues you have, mention it in the chat or the Q&A. But I will do a quick overview of how we've laid out our data in BigQuery. This was a question asked a little bit earlier. So we have a few tables hosted on BigQuery. And to avoid redundancies, we've split them out into a few tables. So for example, here we have our data on the bottom, and then these are all metadata on the top. So tables like compound info and genetic pert info, they contain information relating to genetic perturbagens in this case, and compounds in the compound info case. So these relate values such as like our PERT ID or CMap name to things like target, MOA. Inchy keys and smiles are also available in the compound info and relevant. And these map to SIGINFO files. So there's also a CMap name column within the SIGINFO file and InstInfo file. Similarly, we have.
Sorry to interrupt. Could you try and increase your font a little more? I think the people are still finding it a little hard to view.
Mm-hmm.
Okay.
The tutorial demo notebook, there was a question about that. I'm gonna post this. Yep, so there's a link to the documentation, and again, we can go to tutorial notebook here, and it's also available on the links workshop GitHub repo. Okay, yep, so the InstInfo file is our metadata that relates to level 3 and level 4 data. So here, sample IDs are unique, and there's a one-to-one mapping of sample IDs to column IDs in both level 3 and level 4. Similarly, for SIGINFO, this relates to level 5 data, and there's a one-to-one mapping of SIG IDs to column IDs in level 5. We use gene IDs, which are integer values, as our row IDs in this schema. So that's all available in GeneInfo, and some of the more commonly seen names are listed under gene symbol. So again, this is just an overview of some text related to this, of all of these tables, but we won't go too in depth about, I won't read all this now, but some highlights are, the SIGINFO table includes things like experimental parameters like time point and dose, cell type that was, or the cell line that was used, and also includes some bioactivity metrics such as transcriptional activity score, which you can read about on Connectopedia as well. So hopefully people have gotten this up and running a little bit. So after we have this loaded in, there was a question about how to get the addresses for these various tables. So there is one function here called list tables, and we can run that. And here it says for the cell info table, there is the full BigQuery address for that table. So if you plan to use the Google BigQuery console, this is a useful function just to find where the addresses for these tables. Further on, you can look and get the schema information for a specified table. So here we run it, and we'll get a layout of all the potential column names that are queryable, as well as the data type that's available, right? We can also run raw SQL queries from this toolkit. It's not the main way that we use it, but there is a run query option, and people can write their own SQL query similar to what was written on the metadata browser that Ted showed. And here we again pass the client, which is a common theme, and then our query, and then convert it to a data frame. So there's information about how BigQuery works and the types of objects that are returned on the BigQuery documentation, and the SQL syntax is available through this link as well. Okay, so moving on, we have a few functions. So I've split it into kind of data preview and data retrieval. So these functions allow us to just quickly look at the unique compounds. So they're just useful as previews. They're taking a little bit longer than usual, mainly probably because we have a lot more traffic than usual. But here, essentially, it'll return all the unique compounds available in the compound info table, as well as unique targets that we can, for example, check to make sure that the one of interest that you're looking for is available in our database. And then we have our data retrieval functions. So here, what I've done is I've listed the function name. This is all within the CMap query subpackage. And then here is kind of the documentation right here, and then an example of how to use it. So in this case, we're running CMap query.CMap cell, which will query the cell info table. We're passing it the BigQuery client, which will be necessary for all of these functions again. And then I've listed out some of our core cell lines. So these cell lines have been most abundantly profiled. So we can pass it a list of these cell I-names, and it will return a table of these cells. And here you can see what sorts of data we host on there. Things such as cell lineage and primary disease are particularly of interest. Yeah, and then again, we have the same sort of system with CMap genes. So again, please follow along if you can. And here we have a list of gene symbols, for example. So we pass this gene symbol list to this gene symbol keyword argument. And there are all sorts of other parameters like feature space. So here we'll run this, and this is including all inferred genes. We can also subset this to just get the feature space landmark. So one analysis, one way you can get like all of the landmark genes is to actually run this, and this will give you everything. All 978 of our landmark genes are available with ensemble IDs, gene titles, gene symbols, etc. We also have, again, another one, which is the CMap genetic perts. So this relates to our genetic perturbagens available. Many of these map to the same gene symbols or gene IDs that we have in the gene table. And here, we're again running CMap genetic perts. We can pass it a list of gene IDs, which is here. And then we're returning a sample of that output. So for example, you could see for EGFR, we have CRISPR, shRNAs, and overexpressions of that one gene, as well as kind of a description of whether or not it falls within the landmark subset or the Bing or best inferred genes subset. Okay, so moving on, another one, I'll just expand this table of contents. So here's all the functions, so you'll be able to run this in the future as well. Here's a query of the compound info tables. So here, what we're doing is we're passing it the MOA MDM inhibitor. We can also pass it just this EGFR inhibitor option. Feel free to play around in the Colab space. And when we run that, we get a list or a data frame of compounds as well as their targets and smiles and entry keys of compounds that we have annotated in our database as MDM inhibitors or EGFR inhibitors. Just one quick note is we use, everything is joined with an and operator. So for example, if we have a CMap name, for example, we can put sardemetan. It will do kind of, it'll look for something that has the CMap name sardemetan and is an MDM inhibitor. So it's sort of the most specific results available, right? All right, so moving on, CMap profiles. This is a function that maps to our inst info file from this metadata overview that I talked about earlier. So this responds to individual experiments that have run, so usually at the well level. And here we're looking at sample IDs are the function, are the column that maps to the column IDs in the level 3 signature. So this is a very useful function for being able to retrieve level 3 data. So here I have a list of CMap names. So we can get all profiles available from that were profiled with one of these compounds, and we can do that by passing it with CMap name as well. If you also have a list of sample IDs that you have from some other experiment or are looking at, you can pass it a list of sample IDs and you will get a data frame of those sample IDs. But as you can see, all of the CMap names here are within this list of CMap names, and you would, for the future, you would get, for getting the data back, you would use this sample ID field to query the matrix, which I'll get to in a moment. CMap sig is just like CMap profiles, except it targets the sig info file. So that's collapsed replicates. And here we can run the same thing. And again, you'll get level 5 signature metadata from this, right? You can also, so for example, here you've seen I'm also subsetting by the core cell lines. So these, within the cell iname, it's limited to just those 9 core cell lines in this sample here. Okay, and finally, the big function for, this is for retrieving the actual numerical data. So CMap matrix allows us to retrieve the data from data levels 3, 4, and 5. The way that we have it hosted in BigQuery is actually long form rows. So everything is is column ID, row ID, and then a value. And this function will retrieve those column IDs and then also manage pivoting that data frame into a matrix and return out a GC2 object. So you can read about GC2 in Connectopedia, and there's also a package called CMap Pi that helps interact with GC2 objects. But essentially, that's just a Python representation of a GCTX file if you've used a GCTX file. So here, we've taken a list of column IDs. So here, we've just taken the SIGINFO table that we calculated earlier, and I took some random sample of 10 of them, and then took the SIG IDs and made that into a list. So here, I believe you can just, hover over, and you'll see here's a list of all these SIG IDs that are inside of this list. And we'll pass that to the CID field. We've also stated that we want just landmark feature space. So this will only return 978 genes. And this can be changed to Landmark, Bing, and AIG, right? So here, after running this snippet of code, we get a GC2 object, which has a data df attribute, which actually includes the numerical metadata. So here you have that metadata, and you can do whatever analysis you want to do from here. So this was, again, just a tutorial of just the available functions of the CMap BQ toolkit. From here, I'm going to go into the demo notebook, and the demo notebook will talk more about how to use these functions and how you would go from a MOA of interest to comparing how that MOA performs in various cell lines. So an example workflow such as that. So just to go back to this slide, so if everyone can get back to this this page, find the workshop notebooks, which are here, and then now we're going to move over to the demo notebook. So again, this will require the same setup. So I'll do it along with you here. We'll go to a new tab, CMap. Right. I hope this is big enough. So we'll go to using CMap's BQ toolkit, and then under introduction, we're now going to start with the demo notebook. Okay, so again, we're in Colab. So this is a little more extensive of a notebook with a few analyses. So again, we want to pip install the package CMap BQ. We'll run this code, run our imports. I'm going to go through and upload my credentials file. So that's available. But again, if you don't have that, just uncomment this line, both of these quotes, run this, and then maybe comment out this credentials file path and make sure you're pointing this setup credentials to that JSON, right? So I'll copy the path here in the file browser, and then I'll paste that under this credentials file path variable. And then we can run this. So we have our client, and then we'll move forward from here. So this was covered in the tutorial notebook. So we'll kind of gloss through this, but we'll start right here at analysis 1, comparison of MOA concordance across various cell lines. So the goal of this first analysis is just to show how, given an MOA of interest, we can look at how we would get signatures related to this MOA, find what compounds we have that are related to this MOA, and then get the numerical data, and then also do some correlations and other analysis on that numerical data, right? So first, we can run this first cell. Again, we'll just be kind of running through this sequentially. Here, we want to set, as an example, we have glucocorticoid receptor agonist as our MOA of interest. Within our CMap compounds function, we pass it that MOA of choice. And then we can run this cell. And here we're getting a sample of just a sample of the data frame, but glucocorticoid receptor agonist MOAs and a selection of compounds that are related to that, right? Here we also see that it's targeting this NR3C1 gene that we can use later in our gene mod analysis example to see how much that one target has been modulated. But anyway, moving forward, we'll see a list of, we're able to get a list of how, a bunch of compounds that are annotated with this glucocorticoid receptor agonist compounds, right? Now, yeah, are we able to get to this page? I'm not able to monitor chat while doing this.
Yeah, we should probably just ask if there are people who've been able to get this far, and if not, you should just post questions in chat, please.
Yeah, I can pause for a few moments and then just monitor the chat and see if there are any questions, and I can answer those questions.
Yeah, it looks like there are at least a couple of people who've said that they could.
Okay, that's good. So Anoop, maybe just a couple of minutes for a few more people to try it, because there's a lot of information.
Okay.
So Anoop, how can someone get a list of all MOAs that one can type into this field?
Yep. So there is that in the documentation notebook that I showed. Maybe I moved too fast. There is a list CMap MOAs function, and what this function returns is a data frame of MOAs that are available to put into this function, as well as a count of how many unique PERT IDs are in that MOA that we have on record, right? So
okay, and we should also just make clear that these MOAs are what have been curated from the literature.
Yep.
So that obviously comes with some caveats of its own.
Yeah, I think there was a question about where drug bank and so forth. So I think this might be a good point to point out that in order to get these MOAs, we source them from various public sources. So so drug bank was one of the sources, but we used others as well.
there's a question in the chat about the differences between profiles, signatures, and MOAs. Rajiv, do you want to answer?
Yeah, sure. So the profiles are what we are pre-differential expression, right? So these are normalized gene expression data that kind of maps to our level 3. The signatures are the level 5 differential expression data that have been summarized across replicates. And the level 4 data are the instances. I refer to them as instances. So these are differential expression, but at the per replicate level. That clarifies things.
Okay. So it seems like a few people have gotten to this point. I can try to go a little slower, but maybe it's good to kind of keep plugging on. Okay. So just going back to where we were, um, we queried the CMap compounds function, which looks at the compound info table for compounds that have this MOA that were annotated as glucocorticoid receptor agonists, and we got a list of PERT IDs and compound names that we can now carry into analysis for getting level 5 signatures for, right? So here a little bit lower, this was just a sample of 10, as you can see here. But getting all of them, there are a larger number of these compounds with this MOA. And we can do something similar with the cell line information. So here we're using the CMap cell function. Again, I just wrote down a list of cells of interest. So these are our core cell lines. And running it like before, we're passing the cell line name of this list of cell names, right? I've also added this verbose equals true flag. This is useful for debugging, or if you want to use the metadata browser or the BQ console. This will return, it'll print out the actual query that is run in Google, run on Google BigQuery. So if you want to map how the function, how the CMap BigQuery toolkit is working to how you would use it on the BigQuery console. This verbose true flag is available in many of these functions so that you can see what the actual query is being run. So hopefully we'll get here. So this is the same table that we saw in the earlier notebook, where we can see things like cell type. We can see there's mostly tumor cancer cell types here with one non-cancer cell type. And then there's a variety of cell lineages available. So these are all useful and should all be queryable. So we could get, for example, all lung cells that we have available, or all skin cell lines available, right? Okay, so moving on with those two lists. So we have a list of sample compounds from that MOA analysis earlier, as well as our list of core cell lines here. What we're going to do is we're going to look at our SIGINFO table and try to get all records from that SIGINFO table where the compounds were one of these glucocorticoid receptor agonists and was profiled within one of the core cell lines. So this can be done by just passing both of those keyword arguments. And this return fields all will just give us all the fields. So by default, it's we hand out a priority set of fields. That's just to reduce data transfer costs with BigQuery. But if you need a little more information, then all fields might provide some columns that you were looking for, right? But in general, we have most of the experimental parameters, the transcriptional activity score, replicate correlation, all of that is included in the default priority fields. So this is really more of a niche use case. And then from this sample, we're just doing a few filters. So we have this end sample field. So we want to look at samples that have at least 3 replicates here. We're saying that we want our dose to be at least 10 micromolar, greater than or equal to 10 micromolar, and we're subsetting only so that the time point is 24-hour here. And then this will show a just a sample of 5 of these records. And the SIGINFO file is quite extensive. So this has a lot of columns to it. So there's a lot of information in this table, but some of the more interesting ones are things like transcriptional activity score and replicate correlation. CCQ75 is here. And I think we'll be able to release a table of what these fields are with some description of what the column header means. So I think that we can make that available on the GitHub repo after the workshop. So with the SIGINFO table, you know, you can just jump to getting the numerical data, but I just want to highlight that it is useful to you are able to do some analyses just with this SIGINFO table. For example, transcriptional activity analysis. One analysis we do a lot in, for example, screenings, is we compare our transcriptional activity score across some experimental parameter, right? So in this example, I've taken this SIGINFO file that's been loaded from BigQuery, and then I've created just a box plot of this transcriptional activity score across various cell lines. So just kind of with some a priori knowledge, we know that the receptor related to this MOA is more highly expressed in lung cell lines. And here we have it colored by the cell lineage. And we do actually see that the transcriptional activity score is higher in A549 and HCC515, which are both lung cell lines. So again, the SIGINFO file can be quite useful for looking at various parameters and comparing the activity that we've found within those various parameters. So this can be done with cell line to figure out what sort of cell type you're interested in using, or like time point, or finding the best dose. You know, so the SIGINFO file can be useful using those fields. But then moving on, we can also get the numerical data using the SIG IDs. So I'll just pause for a few moments.
So Anoop, you should point out here that the SIG IDs are the key identifier that one uses to extract information between various sorts of analyses. So imagine you do one sort of analysis and you want to pipe those results into something else. The key field you need is often the SIG ID, right?
Yep. And just to be a little more specific, yeah, our SIG ID is the key to level 5 data. There's another, in the instinfo file, for example, we have it's called sample ID. But yes, this is the key that relates the metadata to the numerical data. So what I'm doing here is I'm collecting all the unique SIG IDs from the SIGINFO table that we extracted. I'm specifying that it's data level 5 that I'm interested in. I'm limiting to just landmark feature space. Again, there are other choices available. And then I'm passing that list of signature IDs. And this will return a GC2 object, which will include the numerical data.
No, there's a question on the transcriptional activity score. Do you want to take that?
Yeah, so the transcriptional activity score is, I actually do have a link to Connectopedia in this notebook, but it's an aggregate measure of strength and reproducibility. So I'll open this Connectopedia link. So here we can see, let me increase the font again. So it's a geometric mean of both replicate correlation and signature strength. Signature strength is calculated as the number of genes that were modulated over some threshold, which I believe is a z-score of 2. So these lines sort of show, this is our SC plot, so signature strength versus correlation plot. This is kind of our visualization of tasks. So these kind of curved lines show what the transcriptional activity score would be for that given point. So each point here would be a signature. So as you get a stronger reproducibility, you're more likely to have a high task score even if your signature strength, the number of genes modulated is a bit lower, and vice versa. So it's essentially kind of how far, how both strong and reproducible a signature is.
Yeah, and it's also worth adding that it's a summary statistic, right? Where it does hides some features of the data, but at the same time, we find it useful to, as a screening tool, to both highlight the really strong perturbations as well as give a sense for range of an experiment as to how much change you notice over the whole screening set. So.
Okay. All right. Where were we? So we've gotten our numerical data. So what I did was I just printed out the shape of that matrix. And you can see there were 434 signatures with this MOA, and then 978 here is our feature space, so our landmark feature space, right? And then from here, you can use the CMap Pi package, which I have a link to here on the documentation about familiarizing yourself with CMap Pi if you've never used it before. But it's a suite of tools that help interact with a lot of the file types that we use on the CMap team, such as the GC2 object, most primarily. So that's all available here, and we can, for example, write as a GCTX file. I believe we can even do this in Colab if I give it some file name, and then you could download it from this file browser if you want to do that from Colab, but also from a Jupyter notebook, you could point it to some path and it'll write out your GCTX file or GCT file, right? So I've commented those lines out, but those, you can, on your own machines, you can run this and make sure that's, and then use that GCTX file in another tool such as Morpheus on Clue or something else, right? ICB, etc. So then the next section is, now that we have our numerical data, we want to do some analysis with it. So for our example, we calculated a correlation matrix to see how similar any of the signatures are with each other. Our end goal is to see if signatures within one cell line, for example, are more similar to each other than signatures across cell lines, just as an example. So I've written some functions here. These are really just utility functions. I'm not going to get too into the weeds about how these functions are working. Again, you have access to the notebook, but this is just doing exactly what it says. It's getting the off diagonals of a matrix, and then some functions to cluster a matrix, just because that can, you know, it's not clearly simple, but you know, in this notebook you'll have examples of how I did it. They're just data frames, so you can do whatever analysis methods you would typically use, but this is just one that I use. And so these are just some functions, and then we'll move on to calculating some histograms. So some analyses that we've done is we're looking mainly in this for loop. We're taking from the correlation matrix compounds of, or signatures of a specific signature ID. So I did that by taking the SIGINFO table, and then I'm filtering it by whether or not the cell iname is equal to the cell within this within our list of cells. So again, it's helpful because we can sort of, well, we can't show it here, but this list is all of our core cell lines. So we're just filtering to see in this for loop for all the signature IDs for a given cell line. And then we subset the correlation matrix that we calculated so that we just have it for one cell line, and then we're plotting a histogram of that cell line. So again, this is more of just an example of some analyses that you could run. So you can always, so please try to run the cell if you can. You can more kind of critically look at the code later and email me with any questions, or contact us with any questions that you have. But this was really just to show a different histograms in each of the various cell lines, and see how the pairwise connections of those signature IDs are in the different cell lines. So what you would expect is a bit higher of correlations between signatures in cell lines where that receptor is expressed. And you, in fact, do see that in the two lung cell lines, the correlations are quite high. There's all these histograms can also be kind of plotted as box plots as well. And here we see the same sort of result, where we see the lung cell lines have the highest median pairwise correlations across all the different cell lines.
So there's just a question, Anoop or Ted,
Yep.
on, can you just cover again briefly situations where you would use this Google BigQuery system? I mean, underlying it's the same data, right? So the question is, can you talk a little bit more about situations when you would use Google BigQuery and when you might just want to download the full data set?
Yeah, I mean, again, it's kind of about like how, like the ease of use and lightweight ability to do the analysis. So so in this example, we're only looking at one MOA, right? Versus downloading the whole set of data could take a couple gigs of download. And if you're only interested in a few things, a few gene targets, for example, or one or two MOAs, you know, it may be considered a waste to download the whole, you know, the whole level 5 matrix and then go through that filtering yourself and having to store that all on disk. So this is really just so you can have on the fly access to the data stored in BigQuery.
Okay, and a related question is, so when we update the data sets, are we going to update the Google BigQuery data also? And how is that versioning going to work?
I think that's the plan, yeah, that we will be updating it. I'm not sure, Ted, do you know about what our versioning plan is? Okay. Well, yeah, so I think the plan is that we'll keep these data stores up to date and just keep, yeah, we'll update them regularly. I'm not 100% certain how we're going to track which versions each one is, but we can make sure to get
We'll
that
communicate that,
out.
but
Yeah,
essentially, obviously, when you query Google BigQuery, our default thing, you'll always get access to the latest data set. If you're downloaded a data set, you're gonna, of course, have to come back and make sure that the one you're working with is still the one that is our current release with whatever updates and tweaks we've made. So there's a bit of a trade-off there. Google BigQuery, you don't have to worry about that. You always get the latest data. But of course, download has its advantages too.
Okay, moving on. So this is just me creating some sort of color map configuration, but next we're going to move on to a bit of heat map generation. So what I quickly did was I clustered all of the correlation matrix. And here, this is a kind of great result. I'll zoom out just briefly, but and here we can see across all of these, 400 signatures, we see a strong cluster here. And these SIG IDs do give some insight to what the profile contains. So here you can see A549 is in the signature ID at 24 hours, and given a certain, there's a perturbagen ID here as well. So these SIG IDs aren't random. They do code in information about what the experimental parameters were. So here you see a lot of A549 signatures clustering quite well, as well as a lot of the HCC 515 cell lines clustering quite strongly here. And then there are other subclusters that are a little bit more faint that can be investigated, right? And then just splitting this up by each cell line, we are running this set of code, which is essentially, again, subsetting this correlation matrix by SIG IDs relevant to that cell line. And then we should, in a moment, get a plot that shows the correlation between each cell line. It does seem to be taking a moment. Let's see if anything else is still running. Okay. This one may be taking a little moment. I think some other cells might still be running that I had started earlier, just so that they can keep going. But maybe we can come back to this result in a little bit. The takeaway of this plot was essentially that across the different cell lines, you would see that the expected cell lines had a much brighter heat map, meaning that signatures within that cell line were more highly correlated between the signatures versus one signatures profiled in a cell line that doesn't highly express that receptor. Right. Okay, so just to make sure we have time for Q&A, I'll just briefly go through how to get level 3 profiles from the level 5 SIGINFO file. So this is something that you might find useful. So given a set of level 5 signatures, you might have a bunch of SIG IDs, and you might want to look at other levels of that data. So here there's a few ways that we've mapped the SIG info table with the level 3 and level 4 profiles. The first one is there's a distill IDs column within the SIG info file. This distill IDs column is a concatenation of sample IDs that were profiled in the inst info table. So here is just an example of this distill IDs. It's not easy to see right away. It's not easy to find, but they were concatenated using this pipe delimiter. So it's again not super easy to see here, but we can retrieve the sample IDs from this field by splitting by this pipe delimiter. So that's what this this block of code does here. It splits that, and then this is just a for loop to flatten that list of lists that results from this code. So here we're taking, for every row, we should get somewhere above 3 sample IDs for each of these rows. And here I've just put down the length of the SIG info that we started with, and then the number of sample IDs that we've gotten from it, right? And as expected, since we filtered as having at least 3 replicates, there may be some with more than that, but we do see that there are at least triple the number of sample IDs for that signature ID, right? So another pretty useful analysis technique would be to use the det plates field. So the det plates field specifies what detection plate a profile was treated in. So this can be useful for identifying control wells that were within the same detection plate as that signature. So here, these are actual plate names. So this maps to a det plate field in instant info. And one way that you could get, for example, the DMSOs, or in our case, our vehicle controls level 3 signatures, is we can run, I'll just skip through to the control wells. We can run a profiles query here where we specify a PERT type. So we can query by PERT type. So here we've said we're looking for signatures within that list of plates. So we took out all the detection plates from our inst info table, and then we just wanted to filter to ones that had this controlled vehicle PERT type. And then we can see here there were quite a few. Sometimes we have, I think, upwards of 20 controls on the plate. Here you can see for each cell type a bunch of sample IDs for DMSO signatures. And just like any other signature, you can query the matrix for those sample IDs. So I think that's what was actually taking a while, because we have a large number of sample IDs here. We can retrieve the data using CMap matrix and specifying data level 3, and then providing it the list of control sample IDs. Right. So here again, just in the interest of time, these are just examples for analysis. It's a data frame that you can run whatever analyses that you are used to running. But this was just a way of showing that even in the control signatures, there may be a higher expression of the target gene. So we were looking specifically at NR3C1 in lung cell lines versus something like a breast cell line. So I know that we're running kind of short on time, so maybe it's good to just open up for questions rather than running through every part of this this last second analysis example. And then again, I'll be available to answer any questions, and you can email me as well. My email is available in the documentation under Need Help, and you can also add issues on GitHub about any feature requests or questions about how to use the toolkit. Feel free to use those two resources to communicate with us.
And also, we will be monitoring email and help desk things actively throughout the day. So if anyone has trouble using it later today after we conclude the session, please feel free to reach out and we'll reply as soon as possible. So Anup, one as a question on so the things you demoed were in Python. What about solutions in other languages like R and MATLAB?
So we don't have a, I don't think we have a solution currently for like an R package. One potential improvement to the CMap BQ Python package is actually making it so we can run this from the command line. So this is something that is kind of planned that will happen in the future. We're thinking about being able to run CMap BQ and then just run it from the command line, and then it'll output some text file that maybe you could load into R. I'm not an R programmer, so I haven't done any work to make this for R, but we would like to make it as easy as possible. So maybe creating a command line version of this tool or making that command line accessible is something that we plan to do in the future.
But given that Google BigQuery, that just uses largely a version of SQL, in principle, people should be able to execute that from an R notebook also, right?
Yes, yep. And again, Google BigQuery has its own sort of APIs, ways to communicate with BigQuery and retrieve data. So for Python, it's the Google BigQuery package. I'm not familiar with what it is for R, but they may have something like that where you can submit your own SQL queries. We just don't have the wrapper functionality prepared for that yet.
Yeah, I'll just chime in. There is an equivalent package for R. It's called Big R Query, but you're exactly right, Anup. We haven't wrapped around that yet, but it is pretty usable, and we've kind of played around with it. So for people who are working in R, that's something to look into.
Okay, so in the last 10 minutes or so, our plan is just to open it up to questions. People can stick around, try things, and post questions. Either raise your hand and we will unmute you and you can talk and ask, or just type it into Q&A and we can address it. And just as a reminder, today the focus was on data access, and tomorrow we're going to have a session focused more on analysis tools, in particular queries and other forms of analysis.
I know the raise hand functionality can be a little bit subtle to see, so we can monitor that. if not, maybe we can go over what the outline is for tomorrow as well. I think Ted had a slide on that.
So I think there's a good question on redundancy of signatures. So there are multiple perturbagens, compounds, and in some cases CRISPRs and so on, that we have multiple instances of that we profiled several times. So the question is, how do people know which one to use? Because it's a little redundant to use all of that. So Rajiv, you have a solution for that. Do you just want to talk about that?
Yeah, I think it's a, it's a question that comes up all the time, right? So you've, let's, taking CRISPR specifically, you might have multiple guides that target the same gene, or you might have multiple replicates for the same guide. And the question is whether you would pick one over the other. I think the task becomes easy if you have many guides, and then you would see whether there's some kind of consensus. But if you have 2 or 3, I think the best you can do is some kind of averaging to kind of smooth over differences between kind of off-target effects and kind of enrich for things that are common. So one approach has been the mod-Z algorithm that we've published on, where the basic idea is to look at the correlation between replicates and then weight, and then compute a weighted average. More recently, I've been playing with a different way of summarizing things, which is to say it's really the tails of the signatures that you care about. And so if you rank ordered each of these signatures and picked and come up with a weight for those features or those genes which are which rank frequently on the top or the bottom of the list, then it might help smooth things over. So I call that fusion, signature fusion. And if there's interest, we can kind of have a small discussion on that tomorrow of how that works. I also should add that, I mean, the other kind of application of this fusion method is that frequently you end up profiling the same thing over and over, over multiple experiments. And instead of considering them as separate entities, one can come up with a exemplar or a kind of canonical signature for that perturbation by just leveraging all the data. So that would be another way you could use it.
If there's no other questions, maybe we could talk about, oh, sorry, I see there's a question. There's a question about whether the size of overexpression data is smaller than shRNA data. Does it mean that there's only one landmark gene for overexpression data? I don't fully understand the question, but I can just answer and say that I think we have profiled more genes by shRNA than we have by overexpression, maybe about 1,000 more. But the number of genes that we measure in each of those experiments is the same. So we're always measuring the same set of 978 genes in all experiments. I hope that answers that question.
And if the question also was, for overexpression, we use a cDNA system to vector, and that has only one construct. So you just type that in one time and you make more of that protein, while for shRNAs, you typically take a few shots at it. So you design a few different guides, which is why for shRNAs, you may see 3 or more different guides for the same gene, while for overexpression, you'll typically see only one per gene. Yeah, the link for tomorrow's session is the same as the one you accessed today. Okay, Ted, you wanna just finish up by what you mentioned?
Sure, yeah, thanks everyone for attending today and for all the questions. If we didn't get to answer the questions today, we will try to answer them tomorrow. You can also send an email to our email account at clue@broadinstitute.org, and we'll monitor that for the rest of the day. I know there were a few issues with people having problems getting their credentials to work to connect via the CMap BQ toolkit. So we will try to debug that too, and hopefully get everyone up and running with that. For tomorrow, we are planning to focus heavily on analysis methods, particularly query, as Arvind mentioned. So we had a thought that as a sort of a homework assignment, people could go to clue.io/query, which is the query app, and just try to run a query analysis. So all you'll need for that is a gene set. So just a list of genes that correspond to some biological state of interest. They could be, you could download them from a website like MSigDB or KEGG or Go or somewhere. Or if you have your own data, you could use that as well. But it'd be great if people could just give that a shot so they can get a feel for how it works. And then we'll go into that in more depth tomorrow.
Yeah, I should point out that it's also a way of stress testing the new query functionality. So the hope is that people can actually successfully submit and it would run in some amount of time. But if it doesn't, it'll give us something to work on.
Okay, thanks all, and see you all tomorrow. And we'll be monitoring email and so on, so please feel free to send us a note if you have any questions between today and when we see you tomorrow again.